Hi, I'm Navdeep Chandel. I'm a professor of medicine, biochemistry, and molecular genetics at Northwestern University, Chicago, USA. I'm excited to share with you a recent study that we published. This is the first paper clip. They're meant to be around 5 minutes or so. And this is really the work of Dr. Sung-Hae Han, who's a terrific physician scientist in my group, who has her own lab now. And going forward, most of my lab members are going to do these paper clips. And so what we were interested in is asking a simple question: does mitochondria control lung epithelial cell fate? Mitochondria are well known to generate energy and metabolites for survival and growth, but more and more they're recognized as signaling molecules to control cell fate or function. And just to orient you, if you haven't been thinking about the lung, and of course during the pandemic this has become probably the most important organ, but adult healthy alveoli is made of type 1 cells and type 2 cells. The type 1 cells are cells that do the gas exchange and they bring in oxygen into our bodies. Type 2 are surfactant producing cells, which keep the lung compliant so it can, like a balloon, go up and down. And then, of course, these cells can get damaged during seasonal flu, the SARS-CoV-2 virus, as well as asbestos, the environmental toxin linked to fibrosis. And now you have a choice. Either you can repair by invoking the type 2 cells as a progenitor population, or you don't repair, especially in the elderly, and you get scarring as fibrosis or respiratory failure, which can ultimately put you in the ICU for long periods of time, and you can succumb to, unfortunately, to death. So for today's talk, we were interested in lung development, on how mitochondria control lung development of these epithelial cells. And the way we went about it is we knocked out mitochondrial complex 1 in the respiratory chain. So if you knock out complex 1, you don't make ATP, you don't do TCA cycle metabolites, you primarily rely on glycolysis. So one prediction would be the cells would just die, right? And we did this in development using surfactant protein C-Cre, which comes in these lung epithelial cells, and we knocked out NDUFS2, which is a catalytic subunit of mitochondrial complex 1. I'm very thankful to Dr. Jose Lopez Berneo and Len Gao and his lab to providing us with these floxed alleles. So we conditionally knock out mitochondrial complex 1. What do we get? The mice die, and they have low lung compliance, right? But the mice don't die in utero. They actually look pretty healthy for the first 10 days, and they're histologically, they look healthy as well. But over time, they start to succumb to aberrant pathology and then ultimately death. And so the way postnatal alveologenesis occurs is, as the lungs are developing postnatally, is that the type 2 cells continue to become type 1 cells, and they go through a transitional cell state, and there's an adaptive integrated stress response that helps them get through that state. And so when we looked at day 21 now, so the mice died day 50, there was an accumulation of these transitional cells, and these transitional cells have parts of have molecular features that look like both type 1 and type 2 cells, right? So now we're getting stuck in a developmental state. Right? And you can see by single cell, these transitional cells have a very high integrated stress response score, right? So they are having aberrant integrated stress response. And just to orient you about the integrated stress response, is designed to get you through a particular stress. So unfolded proteins are made. PKR as a kinase that looks at viral infection. There's GCN2 detects amino acid deficiency. HRI is the one that's really linked to mitochondrial stress. And this will phosphorylate EIF2 alpha, and you'll get a decrease in translation, but at the same time, you'll get upregulation of ATF4 and 5, and this will activate a whole bunch of metabolic genes to get you through the stress. Now, an important feature of this integrated stress response, like all the stress responses you can think of, is either if you can, you have to activate them to some optimal level, but you can overshoot and keep them hyperactivated, and that's not good either. So and there's a compound that Peter Walter's group has generated, which is called ISRAB, which will down-regulate this maladaptive ISR response. And we thought, well, we're seeing such high levels of ISR. What happens if we give ISRAB to our knockout mice, which die typically by day 50, 60? Remarkably, 70% of these mice were alive. Now, think about this. We're not fixing the bioenergetic defects. We're not fixing anything around mitochondrial complex 1 or mitochondrial function. We're fixing something downstream of it. And this allowed the mice to survive now. one of the things we've been thinking about is what's triggering this. So mitochondrial complex 1 regenerates NAD. And so when we knock that mitochondrial complex 3, we got a similar phenotype to complex 1. But complex 2 is parallel to complex 1. They both dump electrons into the Q pool. And complex 2 loss does not interfere with NAD regeneration. And again, we did the same sort of thing in utero, surfactant protein secrete with complex 2 conditional knockouts using SDHD. Again, I'm thankful to Dr. Boneo and Sevilla for these mice. And we got we got some pathology, but nothing drastic. And the mice actually survived remarkably. So they look very dissimilar to complex 1. And really the take-home message here is that type 2 cells, when they become type 1, they go through a transitional cell. But we think that mitochondrial complex 1 dysfunction or maybe 3 dysfunction causes in high levels of the NADH-NAD ratio, which then triggers the pathologic ISR. And if we can bring that ISR down with something like Istrum, we can allow the proper differentiation from transitional cell to type 1. Why the pathologic ISR prevents this differentiation, we don't know. We're using other tricks to definitively show it is the NADH-NAD ratio that's triggering this high levels of ISR, and how it does it are future questions. So thank you very much, and hope many of you contributed to these paper clips.